So welcome back. This is our second webinar exploring discrete genomic results reporting using the Epic Genomics Module. Again, I am Jim Chen. I'm a practicing medical oncologist, and I also serve as SVP for cancer informatics here at Tempus Labs. And as many of you guys know, Tempus is a world leader in next generation sequencing and other advanced diagnostics, as well as EHR integrations. And we're trying to be the leading AI-enabled intelligent diagnostic platform in the world. So kind of a lot of words, but the real focus today is on the amazing success story that Vanderbilt University truly is, both as a university, but also as a precision medicine program. And to tell us about all the exciting stuff that they're doing, I'm really excited and pleased and tickled to be able to introduce Dr. Ben Ho Park. And Ben is the incoming director of the Cancer Center. So congratulations, Ben. That's really awesome, well-deserved. Also, my friend and colleague, Travis Osterman, who is the director of the Cancer Clinical Informatics for the Cancer Center, and as well as Patrick Mergler, who's our VP of Connectivity for Tempus. Welcome, gentlemen.
Thank you.
So really, we're kind of breeze through a lot of the agenda items today. I'm going to talk a little bit about what connectivity is and what the Vanderbilt story is, kind of the vision of precision medicine, and a really interesting, amazing implementation of using some of the discrete genomic elements to power patient care. And we'll talk a little bit about the future. And I'm really looking forward to the Q&A. So just to kind of get us going, again, as you can see here, we generate insights at scale. Tempus has a lot of data, but all this data is really meant to help patient care by aggregating data and finding insights that can be delivered back to the patients. Tempus has over 1,700 employees, and our molecular testing is used by over 4,000 oncologists. More telling, we are connected to over 750 healthcare institutions. And in fact, we're connected to over half of all the academic medical centers in the United States. So, and at Vanderbilt being one of them, but again, we're excited to work with everybody, and collaboration is key. And that's the only way that we can harness data. So that's really unlocked by everybody, so that can be used to help our patients and to help further science. So when we talk about unlocking data and what that means, I'm going to turn it over to Patrick, who will give us a little bit of a primer on that again. This is not the focus of this talk today, but just wanted to kind of get everybody up to speed.
Yeah, thanks, Jim. So just quickly, as a way of kind of introducing the topic, I just want to quickly summarize to the viewers, what it means when we refer to discrete genomic results and the interaction within specifically the Epic Genomics Module, as we'll see here in the experience with Vanderbilt. So since the early incorporation of kind of precision oncology within clinical practice, we all have experience of data being delivered predominantly as faxes and PDFs. And, unfortunately, as we all know, data and PDFs are locked up, not really something you can easily search. You can't do automated clinical workflows or automated clinical decision support. So when we speak about discrete genomic results, what we're really talking about is the transmission for each individual gene and the relevant information about that gene mutation as individual kind of data points or data elements going directly back into the EMR. And so for the Epic institutions, this container in which that genomic data is, kind of held and resides is technically called the VAR. And so, further, this data can also be accessible to trigger workflows, automation of clinical decision support when the institution utilizes the Epic Genomics Module. And so one of our most exciting projects I've ever been a part of that my team has done here at Tempus was we were the first NGS lab in the country to go live with really delivering this discrete genomic results back into Epic's VAR. That project was a presentation at the Epic user group meeting back in 2019. Back over to you, Jim.
All right, so one of the questions I really have is first directed at Ben. So if I were to be hired as a sarcoma medical oncologist at Vanderbilt about 8 years ago, what would have the state of precision oncology been during my orientation? What would you have been telling me?
Well, I wasn't here, so there's caveats to that. But I think when I got here in 2018, it was probably pretty similar. And I think that this is kind of very, very akin to where I think most academic medical cancer centers are today, including my old place, where there's no kind of uniformity or harmonization in terms of how providers are deciding what test to order, which test to order, and then how do we actually get those results back. And what do I mean by that? Well, multiple vendors. Back if you were hired, Jim, several years ago, we had Snapshot as our first next-gen sequencing effort. Another thing called BRL Live for bioreference. I actually forgot the Bioreference Laboratory Inc. I think, Foundation, Guardant, etc., as well as some efforts here and there, kind of one-offs for internal sequencing and for specific types of mutations. Our electronic health record, we have Epic. We had no real integration with any of these results, so we could receive some raw research data, but it wasn't, again, harmonized across all the different vendors. And even if we got the results clinically, it was usually just through a fax, and so someone would have to scan it in and put it in the media tab. And whether or not a provider even knew of that or saw it was kind of hit or miss. There were some molecular tumor boards that were started by some of my predecessors here, but it wasn't kind of the molecular tumor board that we started here, or when I got here that we really started. It was more about an education, almost like a CME thing about, hey, there's this kind of cool case with this mutation, and here's what we decided to do, and here were the results. And while that has value, what I really want to do is put some meat behind molecular tumor boards, meaning, hey, we have this problem, we have this next-gen sequencing data, what should we do, what would people recommend? Germline genetics was really not part of the precision oncology effort back in the day, so to speak. And we also had report storage. I'm not sure what the report storage, but it was unclear if the genomic reports, as I said earlier, were scanned into their into the EHR, and if they were in media tabs. And everyone who knows Epic knows that that's kind of an unwieldy process to kind of data mine. In terms of provider access, there's no method or pathway to ensure that providers knew of actionable results. I think many are aware that this is a relatively new field, and we're not trained in this traditionally in HEMONC fellowship programs. And so knowing what is real and what is not real in terms of actionability becomes a challenge. And then in terms of data access, there's no way of collecting or searching clinical data, as it was not really tied to the genomics data in any discrete elements type of way. Next slide, please, Jim. So where we are today, I don't know if Jim, if you wanted to ask the question if you were hired in 2022, but
yeah, hypothetically speaking, if I decided to, move to warmer pastures, as it were, what would it look like now?
I think in the 1.5 years since I started, and Travis and his team also deserve huge kudos for this, we've come a long way. Rather than multiple vendors, which we still do have for certain indications. Tempus, we've partnered with them to really become the predominant test for all of our NGS testing, not only tumor, but also for circulating tumor DNA, and really more and more germline testing or verification as well. We now have the Epic Genomics Module, which you'll hear a little bit later from my colleague, Dr. Osterman. It's integrated with an Epic seamlessly. Orders go directly from Epic to Tempus, as well as our path support people, and then the results come in as discrete searchable data elements. We now have 2 molecular tumor boards, a weekly one here at the Vanderbilt-Tempus Cancer Center, which we call Big Hope. I won't go into what all these acronyms mean, and then an international effort that we call Our Cancer Genomes, which was using Zoom even before the pandemic. As I've told everyone, I should have bought stock. And then germline genetics has become an integral component of our precision oncology effort. Our genetic counselors and geneticists discuss cases with us on a routine basis, and I actually go to a lot of their case conferences as well. So this is something that has really evolved from the initial efforts when I got here to what it is today.
Hey, Ben, so with the discrete genomic elements now coming in, do you feel like the genomic literacy has improved among the faculty, and particularly among the trainees, because we don't get, as you mentioned, really don't get any sort of education in this?
Yeah, I think there's multiple aspects of it. So the short answer is yes, Jim, but I think not everyone's going to come to every tumor board, and not everyone's going to order genomic tests on every patient. And so there are certain groups, as you can well imagine, like lung cancer, and their patients get this all the time. So that actually, I think, has broadened the kind of visibility, as well as probably the familiarity and education, really, to that point, for lung cancer, the lung cancer docs here and the providers as well, and those fellows that are interested in it. I think we still have room to improve, and I think the educational component is a huge one for us as well.
Perfect. So Travis, I'm going to tap you now. Yeah. Oh, I'm sorry, Ben, you have one more slide here.
I forgot about the continuation. So going on with the comparison, where we are today in terms of data receipt and storage, all the Tempus, and in fact, even some of our other data, which we'll probably get into later, is now in Epic, and that electronic data populates as an inbox result. So just like you would order a CBC, if you order Tempus and those results come back, it's in your inbox message, and you can review it in real time right there, and it's searchable because it's discrete data elements. I'm doing a task right now, which we're trying to move towards automation. Travis will probably talk about that more, that I look at every result that goes out from Vanderbilt, so that if there is an actionable result, I can actually communicate. And getting to your point, Jim, I think more and more people are getting more familiar, even as new indications come out, because I kind of email and pummel everyone and tell them, hey, your patient has an actionable result. We've hired additional staff, so we have a dedicated histotectician librarian, and we've been mentoring this, and that individual has really increased our volume that we have in terms of getting it out in a shortened turnaround time. This is our actual tissue samples to Tempus. We have point-of-care germline testing now being implemented. We have found, and we're going to report on this, many incidental germline findings in our patients thanks to the Tempus germline algorithms and filtering. And now all of our data in Epic is searchable, as I said earlier, but we also still receive separately the raw data in a research bucket.
That all sounds really super amazing. So just to double click on this really quickly, in terms of your histotech, what would you say your turnaround time for Vanderbilt is by the time from requisition to sending it out?
Right now, so it had been somewhere like anywhere from 5 to 10 days, and now we're actually pretty good about 2 to 3 days. And this is something, again, because this person is really bird watching and bird dogging, whatever the phrase is, making sure that when an order gets in there, she sees it and then she really goes after it.
That is all truly amazing, and it's all built on kind of this infrastructure that Travis, I'd love you to talk about, and getting a little inside baseball, into the weeds, whatever you choose your analogy, but take it away here.
Sure. We barely let Ben unpack before we pulled him into really an enterprise-wide group that looked at what precision medicine 2.0 would really look like at Vanderbilt. And one of our challenges was, if you're familiar with the Ackoff model from data to wisdom, we were very interested in some of these higher level concepts around decision support and population health. But as we got started, what we realized was all of these were really predicated on having structured data. And this ended up really being our challenge, that we didn't have structured data. And that became a real focus early on in 2019 and 2020 around the genomics module. Maybe next slide, Jim. And even before that, though, when we started talking about genomic data, especially with leadership in our organization, it was unclear what different parties meant when they meant genomic data. And so this became really a kind of a level setting slide that we used throughout the organization to describe our genomic data strategy. And so if you take the entire set of genomic data that could really start with a FASTQ file and ends up in some sort of structured way onto a report, that's all genomic data, but those are really kind of different pieces of information. So along the left-hand side, we created the vision that any genomic data that comes into our system, we want to receive it just like we receive a BMP. In 2022, practicing oncology, and I would argue medicine more broadly, this test should be no different than a CBC or BMP. It should arrive in our system and leverage all the downstream uses. So you see that discrete data flowing into our electronic health record, and then ultimately flowing downstream to support both patient care and research. Similarly, we would like those upstream files. And so when those are available, that's a separate pathway. So that needs to be available primarily for research, but again, for our internal germline lab, for instance, maybe those are going to support some informed future calls as well. And so we outlaid that we needed an operational data store for all of those raw data, but largely we're going to talk about the left-hand side.
Hey, Travis, so how are you getting all that? So the IC, XML, JSON, FASTQ, SANS, SPAM, CCS. How are you receiving that from Tempus today?
So today we're receiving that by HL7 from Tempus. So we have a point-to-point connection. We're not utilizing some of the newer architecture. We started on this journey before the Aura network was up and running at Epic. That might be what we would choose today, but we have a classic point-to-point connection So we received that HL7 message directly over that. And I think that might be the next slide, Jim. There we go. So this is what it looks like at a high level. Again, for those that are used to filling out the paper requisition, we have an order that's in Epic that looks largely like the paper requisition. Just about every question that's on the paper requisition is in that order. And that gets transmitted electronically directly to Tempus. And then we receive those results back. And by results, I mean a couple of things here. So we receive the structured calls, so that's calls and VUSs. We also receive the PDF directly into Epic. So you still get the PDF, the full report in the media tab. But importantly, you get all those structured results, which means we can start to build on top of those with that model of knowledge ultimately increasing to wisdom.
And in terms of for your providers, as you're kind of going through that, do they kind of just learn how to do the requisitions, or is there a little bit of training? I was really fascinated by how you guys institutionalize.
Yeah, I really like projects that take very little training. And so I think once we told people the order was there, everyone just started using the order because it was faster than going and downloading the requisition, printing it, getting the signatures. So it made it pretty easy to do the right thing.
Awesome.
And so this is just a zoomed in version of what we see in Epic. And so this will look very similar to the report. If you look under somatic there, you can see that by default, Epic collapses a lot of this information. And so they've been great partners to work with as far as what information by default gets shown. So the KMT2A, for instance, is collapsed. And so you can see that that's the default view. But then if you hit that arrow, you can see the Gal NT12. That's all the information that's available there. If you're Dr. Park and want to learn everything about the test, you can expand that. If you're a community oncologist, you can just look at the top line and usually have exactly what you need, including the callouts you see. It's hard to miss the ones that are pathogenic. So I think the biggest value add, though, was not just getting the data, as far as the report goes back to the oncologist, but really how this fed all of our downstream systems. So I really like this kind of data flow diagram. So I've talked about number one, which went live in July of 2021. That's getting the order out to Tempus. And number two, getting the data back via the genomics module. That flows into Epic's data model called Chronicles. Most of us will never interact with Chronicles outside of directly interacting with in Epic. But because it's in Chronicles, what that means is it's available immediately through our patient portal. So that means our patients can see their genomic results if they go for a second opinion somewhere else, or even more likely, if they go back to their community oncologist after having testing here, they can show those results to their oncologist there, who hopefully has access to other means, but it's a nice fail-safe that the patient has access to those as well. Next slide, Jim. If you go down one more, Chronicles gets loaded into a system called Clarity. If you're in an Epic shop, that I think probably sounds familiar. And because it's there, it allows us to do reports within the reporting workbench. This has been really key for us. And so in the insight here, what you see is a search that I did shortly after we went live, looking for patients with KRAS G12C alterations. So these would be patients that, for instance, would qualify for treatment potentially with sotorasib. And you can see that I can select the gene, I can select the type of variant. And because we're doing this in Epic, it also means that we can see who their oncologist is. And we can finally see when they're going to come into clinic next. And this really gets us to where we want to go, which is not just thinking about the individual patient and the individual report, but how do you manage populations of patients, and how do you manage patient populations when new information comes out that might affect not just one patient that's in front of you, but our entire population of patients that we see across VUNC, across all of our oncologists. Next slide. And then for those that are interested more in discovery or understanding their population, again, these are tools that are available to all of my colleagues within Slicer Dicer. We implemented the patients with cancer data model, which is part of the Cohedo set at Epic. And so that creates a registry within Epic for all patients with cancer. And then with the genomics module, you can turn on the ability to do basically searches based on gene and alteration and then slice that. So I've basically recreated that report that I did in reporting workbench, and then I'm using Slicer Dicer to slice out and show me how many of those patients have for instance, lung cancer versus non-lung malignancies for those KRAS G12C alterations. Again, all things that we can do in clinic. You don't have to download any special software. And then for us, the last pieces are that we actually have a separate copy of the medical record that we use for research called the research derivative, or RD, number 6 below, and then the synthetic derivative, which is the de-identified version, the SD below, that's tied to our biobank, which is called BioView, which you can see on this slide. And because these data flowed as structured data all the way from Tempus through Chronicles to Clarity, it also means they flow into our research systems. And so now if you wanted to look at these variants and cross-reference that with some of our biospecimens in our biorepository, we actually do that now. So this is a huge value add, that we were able to get these structured data really to flow down all the way to our research systems.
That's really amazing. So I just want Patrick to chime in really quickly here. Patrick, in your estimation, how was Vanderbilt's integration and go live? Was it atypical? Because they've done amazing things with it. Was there anything different that was done here?
Yeah, so what we found is that Tempus has gone live now with over a dozen Epic institutions that have achieved kind of this level of discrete genomic result integration. And so at Tempus, one of the things we've been able to do is build an expert team of really Epic certified professionals who work in collaboration and partnership with the institution to accomplish kind of integration success. And in doing that, we've built a best practices playbook and anything that we can do to ensure that a hospital will be successful. Because as they say in healthcare, you never want to be the first because you have the scars to prove it. But what we have observed is that as other organizations really, some of them have this programmatic approach, this organizational structure, this kind of institutional buy-in, like Vanderbilt brought to the table, like Dr. Park and Dr. Osterman had put together and established. And this really allows them to, after the integration is live, to optimize their clinical care experience, their workflows on top of that data. However, there are other organizations we see, and we understand, their IT teams, their Epic teams are strapped with competing priorities and competing projects. They complete the integration project, but then they just are, taken on to the next project, and they really don't have the organizational resources kind of left over to really partake in this ongoing journey and this optimization effort. And so we really have seen, Vanderbilt being one of those, shining stars of having that programmatic approach to really make sure that they're optimizing this data within clinical context.
Awesome. Thanks, Patrick. And so I just wanted to turn this over to Ben a little bit here. So Ben, where do you think, what is Vanderbilt's ultimate vision for precision medicine, given this integration piece and given what we've done so far?
Jim, I've used this moniker a lot, and I really mean it, and I'll stick to it. My kind of hope would be that, our vision is to really democratize precision oncology. And what I mean by that is currently, most of the patients that get next-gen sequencing are the result of the fact that their doctors know about it, that they know about it, but it often is the more well-knowledgeable about their disease or well-resourced that can get to an academic medical center or even a community hospital that has kind of the infrastructure to do this type of stuff. And really in 2022, that shouldn't be the case. I think that really all patients with cancer, particularly incurable and metastatic disease, need to have their genetics sequence as well as their tumor sequence. So the slide here really kind of exemplifies this, where we were using, as I said earlier, telemedicine efforts even before the pandemic. And the pandemic, a silver lining has been that it's actually made it more facile. But telemedicine for our cancer patients and also telegenetics at patients at high risk, and there's going to be overlap there, that people would come in and do a real in-person consult or a virtual consult. I think it's critical also to try to get patients. The direct-to-consumer doesn't mean the testing is direct to them, but have them be aware and knowledgeable that if they have cancer or a loved one has cancer, that this actually exists. And that everyone would then get testing of their tumor as well as their germline DNA. We would hold, when needed, a molecular tumor board and consult service using one of our 2 tumor boards, molecular tumor boards. And again, both of those are virtual as well. And then that would ultimately feed into innovative and pragmatic clinical trials, targeted immune therapies, risk management, and screening modalities. And those blue vertical areas really show also we are obviously a research institution, And so we can leverage all of this clinical data as well as kind of the clinical information with the NGS data and really parlay that into various data science applications. I think on the next slide, we might see a little bit more of some of our initial results using kind of the framework of precision oncology and molecular tumor boards. And again, these are just some of the publications that we've had to really exemplify how we're trying to move the needle in this space to use next-gen sequencing and molecular tumor boards for the betterment of patient care.
This is all really, really inspiring, Ben.
Thank you.
And now, so stay tuned, because I want to know from you where do you go from there? You know, what's the future? But I'll ask you that in a second.
Okay.
So just want to turn this back to Travis for a second. Travis, you had showed us how you had kind of looked up KRAS, the KRAS alterations, and somebody told me that you're working on provider messaging for precision therapy prompt. So what is that exactly, and how does this help cancer patients?
Yeah, so full credit to Ben on this one. So Ben really had the vision that we needed to make sure that we didn't miss any patient, and that includes both when the result comes in, but even more challengingly, what happens when a new drug's approved and those results resulted last week, 2 weeks ago, last year, and the patients could benefit from that treatment today. So in the past, we actually had a data science team that would do these queries in the background when new FDA approvals came around. And this has really been our vision, I think, for at least the last 4 or 5 years, is to make sure that we can reach those patients. Next slide. This is really just an extension of decision support. And I really like this framework for thinking about decision support when it comes to genomics. Typically we're thinking about individual patients, and we're either thinking about recommending someone for testing, so they've met guideline criteria for testing, and that continues to expand, and we give an alert that this patient may benefit from genomic testing. Or if you are getting structured data in, using those data to do things like recommend clinical trials or recommend a pathway-based treatment, et cetera. But I would argue that the challenge we should also take up is moving into that right hand, thinking about patient populations. So when screening guidelines change or guidelines for testing change, how can we identify all of the patients in our population at that moment and come up with institutional resources to make sure that they are aware that they can now qualify for testing. And I think the hardest box to fit is actually that lower right-hand box, which is how do we personalize care for entire patient populations when new knowledge comes around? And I think that, again, the classic example, and this came around just before we went live and these data structured, is sotorasib. So on the next slide, just as an example, again, this is a report that now we can run in clinic. And so instead of this being something where Ben or myself or someone else alerts our data team and says, hey, we have a new FDA approval, this is first in class, can you start doing this query? And then working back with the data scientists to make sure that we get the patients right, that we can identify when they're coming to the clinic, who we think their oncologist is, et cetera. We can now do this in clinic with our clinical pharmacy team largely. And I would argue that this can probably be done in clinic between when I'm seeing patients, which is pretty amazing because the tools are right there in Epic. This isn't going out into another data science platform. It's not doing an SQL query in our enterprise data warehouse. These are tools within Epic, which means you can pull Epic up, you can pull up reporting workbench, you can search for patients with the gene and alteration of interest. You can say that you want to see when their next appointment is, who's on their care team. And when that comes back, you can see then, okay, for each of our oncologists, I can then send them a message within the EHR of the list of patients that qualify for this new treatment. This is a really big lift at a lot of institutions, and it's pretty amazing that now we can do this directly within our EHR with tools that are already available, that many of our support staff already use on a day-to-day basis anyway, to really, I think, deliver the highest quality care. And so really loved Ben's vision for this as an idea. And then we've just, I think, really been able to take it to the next level now that we have all the structured data coming.
So Travis, how many people does this take? So you said you can do this between seeing patients. Did you guys hire an extra FTE, or is this just like your staff pharmacist who happens to be, helping you out? Like, This
literally
would
mean
be
my staff
unheard of.
pharmacist. Yeah, literally my staff pharmacist. So Amanda can easily do this in between patients. And now we get really excited when it looks like we might have a new first-in-class approval, because we really can turn these results around. I mean, same day, probably same day as the approval.
That is truly amazing. So, you know, before the integration, how many people would you say you would have had to tap to try to get this information?
Yeah, at least 2 or 3, and it would have taken probably a total of probably about 20 man hours. And some of that is also time going back and forth to make sure that the query was correct, that the clinical pieces were there. And so now, because we can do that in clinic with the oncologist kind of right there beside the person doing the report, it really has sped things up by an order of magnitude.
Again, like I think that's really truly something that is awe-inspiring, just thinking about how much time savings there is and how quickly you can get the drug to the patient now.
And you can imagine that this same framework could be extended to pathways and other systems where you're trying to make sure that, again, the patient's receiving the best treatment for them.
Absolutely. So, you know, Ben, I said I'd come back to you here. So just looking to the future a little bit, what is in the Ben Hope Park crystal ball of precision medicine?
Well, I think if you go to the next slide, one of the things that when we first got here, we had to build what we just talked about and presented. And so that part is kind of done. And we have a lot of stories that we don't have time to go into in terms of some of the impact we've made on individuals and their families. But as I mentioned earlier, really the next steps really are to push this out and democratize the service. I really have in my future goal is that every patient who has metastatic incurable cancers thinks about Vic, hopefully first to the place that they should go to seek out the opinion for getting next-gen sequencing done on both their tumor and their germline, and then utilizing our content expertise to try to help them out, both in the kind of matching of trials, the informatics in terms of what are the assertions in terms of what mutations are effective against which drugs, or the other way around. And then I really also want this to be a democracy of institutions as well. Even though I feel like we are amongst the leaders of this effort, we want to partner with everyone. We want to partner with community hospitals, with other institutions, big NCI cancer centers, and smaller ones. Because I think the more data we share, and I think this is perfect alignment with Tempus's vision, the more data that's shared by everyone, the better off we all are. You know, a lot of people are doing what I call N-of-1 experiments, but it's never being recorded. But if we can get that into a database that we can all share, and this is a concept that I've talked with many others, everyone wins. So the next time someone has a rare mutation and a rare type of cancer, we can share data and say, hey, you know, someone's actually done that, used that drug before, and they had a response or they didn't have a response. And I really feel like that for me is the future and where we want to be, and hopefully we'll get there in the next 5 to 10 years.
I'm sure you get a lot of phone calls, Ben, you and Travis both. There's so much accumulated knowledge here. It's really, really amazing. So, you know, Travis had alluded to the Aura network earlier, and this is kind of what's down the pipe for some of the integration work. So I'm going to turn this over to our expert here, Patrick, so to speak to a little bit about Aura.
Yeah, thanks, Jim. So, you know, as mentioned before, you know, Vanderbilt, when they went live with this integration, the Epic Orders and Results Anywhere network, or also known as Aura, wasn't really at a position to support broad NGS testing. The good news is it is now. So with some of the recent releases of Epic. And so Tempus has always really recognized the value with labs and institutions connecting in a more automated, streamlined way to support clinical workflow. So when we you know, understood the competing priorities of hospital IS teams and their leaders that they face, you know, given personnel requirements to kind of traditionally build these lab integrations and setting up these point-to-point VPNs and opening firewall rules and information security analysis, you know, it requires a bit of due diligence, you know, months of governance and prioritization, you know, months of actually executing the project. And what we saw, you know, was really an opportunity for us to invest in what Epic calls the Specialty Lab Diagnostic Network. And we did that last year. And in doing so, because we're connected to that Epic ecosystem, this really enables Tempus to connect with our Epic institutions utilizing already existing infrastructure there, the Care Everywhere infrastructure. And under the Epic's Orders and Results Lab Anywhere network, this really dramatically reduces the number of staff, the hospital IS resources necessary, and complete this integration with Tempus in a more streamlined way. Also utilizing standards, so it's repeatable. And since it's utilizing that existing standard, we now have literally dozens of health systems that are, looking at this second half of the year, looking to establish a connection with Tempus in this manner, because they can just roll it into their normal quarterly upgrade process. So it's not a big, huge project that has to be separately governed and groomed and prioritized. It becomes a part of their upgrade. It becomes a part of managing their system. So that's what's really exciting about what this kind of innovative model that Epic really has enabled. And we're really excited about how we can interact and engage and connect with more and more providers and more institutions.
Thanks, Patrick. And this is, again, so many things that are so exciting, so many things down the pike. I feel like the next upcoming year or two are going to be really very defining years for precision medicine. So for those of you who are on the webinar, thank you again. We are taking questions, and we'll try to get to as many as we can. The rest we'll try to take offline. Again, a great thank you to Ben and Travis and the Vanderbilt crew. So I had a question I wanted to ask both. One of the questions that came through here for Ben, for AMCs that are taking steps to set up precision medicine programs, what do you recommend?
I think the biggest, most important component of this is to get leadership institutional buy-in to make sure that there is some sort of support for harmonizing vendors, and you don't necessarily have to just stick with one vendor. And again, we don't have a single vendor, but it certainly does help to stick with one that the majority are using, so that we can actually use that as the initial framework, if you will. But I think you need leadership buy-in to make sure that they're going to support this effort, that they're going to have and hire, if they don't have, hire really capable informaticians, especially someone like with Travis's skill set. By the way, there's not many of them, and so don't anyone poach him from me. And I think that that's really, it sounds easy, but it's not. And I think anyone who knows this space and this field recognizes that there was a lot of heavy lifting in that first year and a half. But again, the precision oncology team, which includes Travis and a lot of his team as well, put a lot of hours in. And there was points where, and Travis knows this too, that a lot of pain points that we had to get over. But I mean, look where we are now. And once you've got that part done, again, we've set the groundwork for it. Now is the super fun part, is just taking off and getting more and more patient sequence.
Thanks, Ben. And so Travis, here's a question that came through for you. Outside of the sotorasib example, are you doing anything for clinical trials feasibility?
Yeah, it's a great question. So I think many folks that live in the clinical trial world can relate to this. And it's a story I hear all the time, which is I have a pharma partner that comes as a disease team lead to one of my colleagues and says, we're interested in opening a pick your favorite target in an NTRK fusion study. How many NTRK fused patients do you see each year? And historically, I think what most disease leads would do is kind of guess based on their experience and their practice. And some of them are probably really good at that. But as we subspecialize more and more, and especially as you're talking about site agnostic trials, it gets really hard to guess for these really infrequent targets, how frequently those come up. And so we use this now to do a service within the Cancer Center that we just call clinical trial feasibility studies. And so any of our investigators can basically ask that question, and we'll turn around and use the structured data to give them a real answer, so that they can make more informed decisions about what trials they open or don't open. Because anybody that's in a CTO knows the most expensive trial to open is the one that enrolls one patient. The next most expensive trial is the one that enrolls zero. And so you want to make sure that you have the populations to see. That's a service that we are leveraging these data for.
Perfect, thank you. I saw another question come through. Will the slides be available after the talk? Actually, this whole presentation is recorded. And so for those who wanted to kind of, pause on one of Travis's beautiful slides or listen to Ben again, Ben's dulcet voice, and in a certain section, that will all be available for you. Hopefully the recording link will pop up right afterwards. If not, please just let us know. And just a quick plug, both Patrick and I will both be at ASCO. There's actually a Tempus booth. We're actually doing a ASCO talk there about some of the work that we're doing right now. So a great time to just check in with everybody if there's interest there. Last question, and then I think this is the last, then we're basically at time. So Travis, very quickly, one question came in. So for all your other NGS data, how are you synchronizing that right now?
Yeah, it's a great question. So, I luckily have had some really great predecessors that worked really hard to get whatever structured kind of format, proprietary format from any vendor that we've ever worked with, dating all the way back, as Ben mentioned, to our internal panel, which for melanoma was just BRAF back in 2010, 2011. So we've taken those JSON, XML, whatever we could get files, and have done the crosswalk into Epic's format to basically create our own HL7 messages as if we were the reference lab, and have been able to fire those against a test environment in Epic and load those in. We've finished clinical validation, and we've got one final contract that we need to amend it, and then we'll enter those into production, and we'll get another 9,000 results. I'm really excited about that process, and hopefully we'll get a chance to share more about that in the future. But if anybody's interested in that crosswalk, I'm happy to follow up.
Again, as always, amazing work, everybody. This is, again, Vanderbilt's quite the leader in precision medicine, and here we can see why. So to thank you again very much for your time. If there's questions from we'll take the rest of the questions offline and try to get back to everybody in a timely manner. If there's questions that you want to ask either Ben, Travis, me, or Patrick directly, our email addresses are there. I'd love to chat at ASCO if you guys are around. So thank you again, and we'll see you for the next webinar. All right.
Thanks, Ben. Thanks, Travis. Take care.
Thank you.